Thanks so much for that introduction. As Dirk said, today I'll be talking about functional anomaly mapping and also perilesional neuroplasticity in post-stroke aphasia. And thank you so much for coming. Thank you so much for the invitation. I am now a C star. Before starting, I'll say I have no financial relationships to disclose. First, let me add a little bit more color to what Dirk said in my introduction. I studied linguistics starting out in Philadelphia, where I also got my my clinical degree in speech, then went to Arizona, studied with Paige and Stephen Wilson, and then finally came to Georgetown to study neurostimulation and got some machine learning experience with Peter Turkeltaub. And I'm happy to say that I am now not instructor or postdoctoral fellow, but assistant professor of rehabilitation medicine here. And as Dirk alluded to, my interest kind of melds both the clinical side of things and the theoretical technical side of things, with a real emphasis on how do we take this information that we're all trying to gather and make it work for us in a clinical context. And when you actually make that the minimum bar for success, you can come to some very strange conclusions that might be unintuitive. But let me just get started. As I alluded to before, I'll be talking about two different projects. We'll take them in the reverse order. First, I'll talk about a project we published on perilesional plasticity in post-stroke aphasia, and then I'll talk about functional anomaly mapping. And we'll hopefully get through everything, but feel free to interrupt at any time with questions, and we'll take them as they come. So to start off, a perilesional plasticity in post-stroke aphasia. So this is a very special group here because so much of what I'm going to talk about is old school and familiar to everybody. So I'll skip some of the basic things, but we know that most of the behavioral deficits that follow stroke are driven by the lesion size and the location. But as we, I guess, all know now and all believe strongly, that there are things happening outside of the anatomical lesion area that are going to be driving how much recovery we see from person to person. And those things that are happening outside of the anatomical lesion are the things that we really need to understand in order to figure out how we can optimize outcomes and understand what makes somebody get better versus not get better. And although there have been, I guess, over the past 20, 30 more or even more years, many studies talking about patterns of changes, for instance, in functional MRI, what we really need to find are mechanisms. And when I say mechanism, I mean, let's take those patterns and let's try to understand them in a principled way and try to figure out in a way that we can kind of understand and maybe someday in the future control to ultimately optimize outcomes. When we think about just the left hemisphere, several different patterns, several different mechanisms have been proposed. The one we'll talk about here in the first half of my talk will be perilesional plasticity, but normalization of broad spread network dysfunction has also been proposed and observed by people like Dorothy Sauer. And then there's several lines of work showing that there may be recruitment of alternative processors that aren't as involved prior to somebody having a stroke, and afterward they become involved. It doesn't say it on the slide here, but a common cognitive domain here that is often mentioned is quote unquote domain general processes, and some of my own prior work has found evidence for that. But today we're going to be focusing on perilesional plasticity and the perilesional plasticity hypothesis. Now, the perilesional plasticity hypothesis puts extra special weight and emphasis on the tissue immediately surrounding the anatomical lesion that comes from the stroke. And the evidence for this tissue being particularly of interest goes back, again, many decades, where animal studies found that the tissue that is structurally spared around the lesion boundaries shows abnormal function, but also shows cellular changes that might be involved in helping somebody, or in this case, an animal model, recover after the stroke. And so these cellular mechanisms are described potentially as collateral axonal sprouting, synaptogenesis. And if you look over here, I hope you can see my little laser pointer. If you look over here, ipsilateral effects from this figure from Nudo 99. These ipsilateral effects are special things that are happening at the cellular and molecular level around the lesion. And in fact, there's evidence suggesting that these changes may also be evident in humans when, for instance, somebody has a stroke that affects their motor system, that there may be a sort of functional takeover by perilesional sensory motor cortex or primary motor cortices when those tissues happen to be around an individual's lesion. One of the big themes here is that the lesion is different for every different person, and that makes doing this kind of study where you're trying to understand perilesional, the role of perilesional tissue, particularly challenging. And so with this study, we had basically, it's kind of a simple aim. What is perilesional activation? And on the left side, with respect to the perilesional, basically neuroplastic recruitment hypothesis, the classic perilesional hypothesis, if that's the case, then we would expect in post-stroke aphasia that around the lesion, activity should be increased relative to controls, and we might find activity outside of the typical language regions, right? If the perilesional tissue is outside regular language regions, well, if it's perilesional and perilesional tissue is important, then we might find new activation there. I won't go into great detail here, but we may, for instance, find increased activity for small lesions relative to large lesions. Maybe a large lesion destroys any capacity to show changes because of a large-scale destruction of tissues. And finally, we would expect that if perilesional recruitment is at play and it is supporting behavior, then we should find the amount of activation relating to behavioral deficit. On the right side, you can see an alternative hypothesis, which is linking back to the Dorothy Sauer citation earlier, which is basically the idea that after a lesion, you have widespread network-wide dysfunction, and it's actually that the network is coming back online as a key to recovery. And if this is the case, then we should find activity that's the same or reduced relative to controls, and we should find that same network come back online, which means that the activity should be in typical language regions. We should find that large lesions are related to reduced activity because, of course, the network is going to stay down and underactivate. And then also, again, we should find activation related to behavior. We also ask the question, are alternative left hemisphere, that is to say non-language left hemisphere processors recruited? And can we see evidence for this in multiple different tasks? So this is basically what we did. And one thing I should say is that typically this kind of question is asked without reference to a healthy control cohort. And one of the things you'll see throughout this lecture today is that it's really important to be comparing the patient to basically a normative distribution based on healthy controls. Okay, so to jump right into the methods, we have two tasks. We have a spoken picture naming task, and we have Stephen Wilson's adaptive semantic decision task. And we gave these tasks to a group of patients and a group of controls. And you can see here's a lesion overlap of the patient group for the naming task, lesion overlap for the cohort of the semantic decision task. And we're basically going to take this data and do what we just sort of alluded to, which is examine fMRI activity during the task in language areas, in what we here call language capable areas, and in non-language areas. And as I alluded to earlier, one of the challenges of this kind of work is that it's easy to talk about perilesional tissue, but perilesional tissue is actually different for every different participant, every different patient after the stroke. And so we have a bit of a methodological flourish here to try to capture that. And so the way we're defining tissue here is to first, in every patient, we're going to trace out the anatomical lesion based on their MP-RAGE, so it's typical procedure. And then we're going to adopt a procedure that's been used before by Julius and other people, Jessica Richardson, basically take that lesion mask and dilate it. So expand it out in all three directions, and use that tissue, that shell around the anatomical lesion, as the definition of perilesional tissue. And in this study that I'm describing, we did two sets of analyses. The first set, when we look brain wide, we're going to use that shell and we're going to expand it several times and look at several different distances from the lesion edge for each of the patients. And then there's some more detailed analyses, and for those, we just consider tissue near versus far, because you need a very large sample size to adequately have coverage of all the different parts of the brain in all these different combinations of tissues. So I'll show you some more pictures and the results in a second, but the first thing to think of is how are we defining language regions and how are we defining language capable regions? So these are the group activation consistency, so penetrance map, what percentage of participants activate in each voxel. In the left column, you can see the naming task results. In the right column, you can see the semantic decision results. The controls are on the top row, the patients are on the second row. And then on this third row, we have the way that we're defining language and language capable tissue. So the red is language tissue. And this is a mask from Neurosynth using a keyword that's related to the task. And we've dilated that, and that's shown in green, and that's what we're operationalizing as language capable tissue. And you'll notice if you look carefully that, I mean, I've been surprised by this, and every time I look at it closer and closer, I'm surprised by it. This independent meta-analytic mask is like pretty darn good at capturing what we see empirically. And so what we're going to do with these language areas, language capable areas, is we're going to intersect those different groups of voxels with the perilesional tissue that's defined for each individual patient. And so here, language tissue is invisible. We have language capable in red. We have this patient's lesion in blue, and we have the perilesional shell, or the first perilesional shell, in green. Overlaps, mix the colors. And so for each patient, each individual stroke survivor, we're going to do a procedure where we map out the anatomical lesion. We're going to dilate that lesion to find perilesional tissue at several distances, and we're going to intersect that perilesional mask with the language mask, the language capable mask, and a sort of non-language tissue leftover set of voxels. And we're going to go into all of the control subjects and grab the activity from those intersected mask overlaps. And what we're going to do is we're going to estimate a large mixed effects model where we try to predict the amount of activity observed at that intersection of mask type and group type, and see, is there a difference in activation in patients versus controls, for instance, in the language tissue when it's perilesional 0 to 4 millimeters, in the language tissue when it's perilesional 4 to 8 millimeters from the lesion boundary. And we'll do that same thing in the language capable tissue and also the non-language tissue. And so we'll jump right to those results. So this is going to allow us to ask, is there recruitment in perilesional tissue above normal levels in the patients? And so here, the top row of panes is the results for the naming task. The bottom row of panes is the semantic decision task. And this first column is the results of that comparison for the language tissue. And the second column is for language capable tissue. And the third column is non-language tissue. And the x-axis on each one of these panes is distance from the lesion boundary, which is to say how far out that perilesional shell was situated with respect to any given patient's lesion mask. And the y-axes show the difference between patient group and control group. And so basically what we're seeing here is that in language tissue, the closer you get to the lesion, the less activity you have in patients relative to controls. So if we were looking, if we were going to find perilesional tissue recruitment above and beyond normal levels, we would expect that line to be up here. But instead, we see that there's underactivation in the patient group around the lesion out to 8 to 12 millimeters in the naming task, and just around the lesion in the semantic decision task. Now, looking at language capable tissue, we see the exact same pattern in the naming task, reduced activation rather than increased activation. Again, this is anchored to controls in those same little overlapping chunks of voxels. In the semantic decision task, there is no underactivation, but you can see that the pattern is very similar. It's below as opposed to above that line. Non-language tissue, very close to the lesion. There's reduced activation in patients and no difference in non-language tissue in the semantic decision task. Okay, so one thing is it maybe that the big lesions are messing up this relationship? If we look at just small lesions, can we find increased recruitment relative to controls? So here on the left side of this slide, you can see again, the top row is naming task results. The bottom row is semantic decision task results. And now we're focusing on just the language tissue. So if we go back one, it's just this left side column. And here we're showing every individual patient as a dot. And the gray outline is the healthy control range. And this left column is showing the results near the lesion. And the right column is showing results far from the lesion. So here we have a data sparsity issue, so we can't use little fine-grained shells. We have to use big, thick slab. And so basically what we're looking for here, if we are to find supernormal activity is in small lesions, is that when we're closer to 0, which is lesion size on the x-axis, then we would want to see the dots go up, 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 and go above that gray band. But in fact, essentially nowhere does a dot go above that gray band. So nowhere are we seeing supernormal activity in these small lesions. And in fact, in the larger lesions, maybe not surprisingly, in those larger lesions, we're seeing a relationship where there's underactivation or reduced activation relative to the control cohort. Now, thinking about whether the amount of activity that we're seeing independently relates to behavior. In this case, we're correlating the amount of activity in each patient with their aphasia quotient. Here we're looking at the naming task results near the lesion, and there's no relationship in that task with behavior independent of lesion size. And now far from the lesion, similarly in the naming task, there's no relationship. Turning to the semantic decision task, there is a relationship in activity, independent activity with aphasia quotient elicited by that task near the lesion, independent from lesion size. And again, far from the lesion, there's also a relationship between the amount of activity in each patient and their aphasia quotient, some behavioral measure of their language ability, independent of lesion size. So that's most of our analyses. We have one more here, and we wanted to say, okay, is there perilesional activity increases, but it's maybe not everywhere in the brain, it's only in a specific region. And so for this, we took the brain, we broke it up into a bunch of parcels, and we tried to do this analysis at each parcel, again, for the naming task on the left and the semantic decision task on the right. And the top row now is results from near the lesion, and the bottom row is results far from the lesion. Basically, if we're going to observe and I should say blue is patients less than controls, red is patients greater than controls. Basically, if we were going to see increased activity that's perilesional in a specific brain region, we would expect to see maybe a red area, that is patients greater than controls, near the lesion, but not far from the lesion. And if you notice, there are some red areas showing that there is perilesional increased activity here, but that activity is also increased when that tissue is situated far from the lesion boundary. And so although we could say maybe some small portion of occipital lobe is showing a systematic increase in patients relative to controls, it's not particularly, it's not increasing when that tissue is particularly close to the lesion boundary. I'm just reading Dirk's comment here. Thanks, Dirk. So to take stock of what we had predicted, or we had talked about a few minutes ago, we see a lot of X's on the left side of this table in sort of not in favor of neuroplastic recruitment, and we're seeing a lot of check marks on the right side in favor of this renormalization of network dysfunction. We do find alternative left hemisphere processors recruited, which are those red spots we saw in the last slide, but they weren't particularly increased when that tissue is situated close to the lesion boundary. And we also found this pattern was essentially the same across both of these tasks. And so to conclude the first half of this talk here, we didn't really find evidence for perilesional plasticity, although I want to see it. We did find evidence for lesion size dependent widespread language dysfunction, and that favors a renormalization or normalization mechanism. And I should say, although these findings give us some information about what's happening in post-stroke aphasia in the brain, some of us are probably thinking, well, these are just a couple of tasks. You've only focused on perilesional tissue, so really you haven't said much beyond that. And I strongly agree. And so in order to go beyond a specific task and beyond only perilesional tissue, I want to pivot back to this functional anomaly mapping notion. And so this second part of the talk, I'll roll out in 3 sections. I'll give you a quick background. I think we could probably speed through that given the group, but we'll say a few things. And then I'll describe to you how to, in sort of schematic terms, derive this functional anomaly map, whatever that means. It'll be clear in a second. And we'll do that for individual patients. We'll get their own functional anomaly map. And then I'll show an example of how we think this kind of functional anomaly map could be utilized. And in this case, it's going to be in the context of lesion behavior mapping. So take a big step back to this idea of lesions. So I always like to mention the Edwin Smith papyrus, but it takes like a long time to follow history all the way up to today. So we'll just say that the lesion deficit correlation idea goes back a couple thousand years. I think that's 1600 BCE, if I remember my speaker's notes. And with a low sample size, people like Gall and Wernicke and Broca have really laid the foundation for modern neurology and kind of what we have come to kind of take for granted in terms of the brain behavior relationship in communication and communication disorders, in particular aphasia. And so in the, I guess since is it the 19th, 20th century, the late 20th century and 21st century, the lesion idea has kind of been overshadowed by the emergence of fMRI and activation studies. And I always use these 3 movies actually as touchstones. So CT, 1974, that's Godfather 2. MRI in 1977, the original Star Wars. At this point, I can't keep track. And 1992, that's the original Aladdin, well, Disney's original Aladdin. And so in this time, the regular lesion method is starting to become overshadowed. Nevertheless, the lesion method has evolved, as probably many of us are familiar, where instead of just looking at what side of the brain is related to, contralesional weakness or whatever, or, frontal damage causes an expressive deficit, whereas posterior damage causes a receptive deficit. With modern imaging techniques and modern computing power, we can go with very high spatial resolution to look for lesion behavior relationships. We have voxel-based morphometry. We have multivariate approaches. We have ways to include white matter. And so the lesion method has been alive and well. By and large, however, the lesion method, whether it's going back to Broca or whether it's doing VLSM, has made these 2 major assumptions. And the first assumption is that tissue that appears anatomically abnormal, functions abnormally. And the second assumption is that tissue that appears anatomically normal functions normally. And although these seem like logical assumptions, there's some pretty profound consequences. So as you can see to the right, I've sort of drawn a red line here around the lesion, but like, is that the right boundary? Kind of looks okay, but what about this middle, dark gray area? Is this tissue functional? Is it partially functional? Is it equally dysfunctional? So lesions so often appear heterogeneous without clear boundaries. So if we trace them out slice by slice, which is what we typically do, then we're forced to make an arbitrary decision about that. And we can't know, based on just the anatomical image, whether that tissue is functioning normally, or if there's any degree of dysfunction. And of course, for over 100 years now, we've known that a restricted cortical lesion can and often does cause remote dysfunction, even far from what you see on the structural MRI image. And so these distant changes can include things that are typically seen as negative, like diaschisis, which is a term that I guess I tend to stay away from because some people really don't like it. It means different things to different people. And I will instead usually use the term remote dysfunction, but also will be the locus, outside the lesion will also be the locus of changes that are a consequence of the lesion that might be good, like neuroplasticity that helps somebody get better. And by making these assumptions in our lesion symptom mapping analyses, our lesion behavior mapping analyses, we're building in the impossibility of these phenomena factoring into our findings. And so I'm not the only one to think this. And so there are other people that are working on this, and there are actually other ways to measure the functional status of spare tissue. The one that is sort of the most attractive and interesting, maybe not the most attractive, but very interesting, is PET scans, where you're looking at some measure of brain function through a radioligand that might bind to some specific interesting receptor that you want to see how it's functioning. But PET scans involve ionizing radiation, they're very expensive, and there are other reasons to just prefer them and make reasons why they're hard to use. Other approaches to measuring the functional status of spare tissue have involved, for instance, task-based fMRI. So like Corbetta's, spatial neglect recovery, Ward's motor stroke recovery. The problem, or some problems with task fMRI here, is that you're and I should say also what I showed in the first half of this talk. Some of the limitations to this kind of task-based fMRI approach is that you're restricted to a single set of neural processes, and that's assuming you've got a task that's reliable and valid. And also, it's sometimes difficult to interpret the results of a task like that in the fMRI signal, because a patient may experience a task in an entirely different way than a healthy control subject, because they may not be capable of doing the task, or they may be doing the task in a completely different way. And finally, there's a family of approaches that rely on resting state fMRI. And so this includes lesion network mapping, where you're looking for the effects of spatially disparate or far apart different lesions to different places that might have some overlap in a connectomic sense. So the anatomical damage may be different places, and the thing that makes them cause the same symptom, even though the damage is to different places in different people, is that there are functional consequences that, if you can measure them in the individual, you may find overlap in space. There's a hemodynamic lag approach. We could talk about ALF here, and there's several other emerging approaches. And so what we wanted to do, though, was we wanted to say, can we develop a method to directly map functional aberration throughout the brain? And in the context that we're talking about, we wanted to hopefully try to remove the need to make an arbitrary lesion boundary. We wanted to try to use that brain-wide map of functional anomaly or aberration to extend the reach of lesion symptom or lesion behavior mapping to do analyses that can basically detect those phenomena like diaschisis or neuroplasticity that extend beyond the boundaries of the anatomical lesion. For instance, if you do an analysis of patients with left hemisphere stroke, and that's your entire sample, it's impossible to find lesion behavior mapping results in the right hemisphere because, of course, you have no coverage there. And finally, more generally, to provide some direct measure of functional differences that can be applied to a single individual patient, because, of course, with clinical translation considerations, it's not good enough to work at the group level. It must work at the individual level. And these kinds of differences might be related to remote dysfunction or diaschisis, some type of plasticity, or some other individual differences. And so that's kind of what we try to do. And that's kind of what I'll present here in the next 15 minutes or so. So what I'm presenting is data from about 50 patients. Each of them are chronic left hemisphere stroke survivors with a history of aphasia. Almost all of them also have aphasia. And what we're going to do is derive a functional anomaly map result image for each patient. And we're going to incorporate as a norm reference cohort, we'll factor in 57 healthy adults who also participated in this study. And so there's the lesion overlap on the right. You can see typical MCA distribution. So each of these patients and each of these control subjects did a resting state scan, and they had anatomical image for lesion tracing and normalization. And it was important for us to try to reduce the obfuscation, sort of make it as simple as possible. And so here we take this resting state data and we apply regular con pre-processing. And notably, many of the patients, because it was retrospective in the lab, many of the patients had multiple scans. You can see here that, I have a little pop-up here. On the right here, you can see that the number of sessions for each patient goes up to, like, one of the patients had 8 sessions. Many of them had 3 sessions. And so this is going to allow us to examine the reliability of these images. So we'll jump right in. So here I'm describing support vector regression, support vector machine regression. It's fancy sounding. It's basically just a multivariate regression. And unlike some newer machine learning techniques like deep learning, it's one of the members of the machine learning family that has a very straightforward interpretation in a regression context. It's basically just like a high dimensional multivariate regression. And most people who use this type of approach, support vector machine regression, they're using the model to make predictions. But here, we're not using the model to make predictions. What we're doing is we're estimating a model solution that separates the patient from the control group. And then we're going to bust open that model like cracking open an egg. And we're going to back project the high dimensional solution into data space. And this is called a pre-image. If you look up pre-image on Wikipedia, your eyes will quickly glaze over, but you may find some interesting remarks on what this is. It's used in cryptography, but it's really described as such. And it's also worth noting that we're using support vector regression, but in my view, it's much more important the logic of what we're doing here as opposed to the specific algorithm. So to say very quickly what we do with the data, for each patient, we're going to compute a single image, a 3D image. And the way we do that is by taking their 4D pre-processed resting fMRI scan. And we're going to, that is X by Y by Z by T. So it's a lot of big data, 2 gigabytes or whatever. We're going to squish it down into a really long vector. And then we're going to do that same procedure for some number of control subjects. And we're going to concatenate that all into a big matrix where, each of the columns is is one of the subjects, and each of the rows is one of the voxel values. The data is all in a standard space, but it's 4-dimensional still, and the nuisance regressors have been taken out. And then we're going to compute a contrast matrix with dummy coding to try to separate out the patterns visible in the patient relative to the controls. And then we're going to back project that model solution. It's a simple multiplication back into a data set, which you can think of as a pseudo data set. The way I think about it, and I think the way I've seen it described, is it's a data set that basically represents the high dimensional feature space mapping back to the real world, essentially. And what we get is a 4-dimensional back projection that also sort of occurs over time. So it's x by y by z by t. And we're going to average over the quote unquote pseudo time dimension. And what that gives us is a 3D norm referenced and normalized, spatially normalized 3D functional anomaly map for a given patient. And it says at each voxel, how much deviation you might have observed relative to the reference cohort, considering all the points in space and over time. At least that's the idea. So let's look at a couple of these. So basically, what I'm showing on the right is several patients, PRO, AFB. These are code names. On the top row, you can see PRO. Here's the anatomical lesion tracing. And you can see I've got these 3 results from 3 sessions here showing basically the functional anomaly map. And here you can see there's no empty voxels, but for the purpose of showing it and describing it here, we're thresholding it to show that here is the highest loading of functional anomaly, and it's loading onto appears to load onto the anatomical lesion. Okay, you can see these are 3 separate scan sessions for that patient over weeks or months difference over time. Second patient, similarly. Third patient, similarly. So it's interestingly reliable. And then this last patient down here, they have a deep subcortical lesion. And what we see is the highest loading not on that spot of deep subcortical lesion, but we see a reliably discovered distributed anomalous signal sort of throughout the cortical mantle, wrapping all the way around, even onto the medial surface of the left hemisphere. This is kind of like what you might think of in a textbook or something. So this was very interesting to us. So we asked 3 questions about these results. The first one is, are we actually detecting the anatomical lesion? And so we used a receiver operating characteristic curve approach to test this idea. Basically, an ROC curve analysis asks, can one thing classify or categorize another thing? And you get this line, and if the line is perfectly diagonal, it's random chance. And if it bends upward, that means it's greater than chance, it's non-random. And so when we ask, can the functional anomaly maps be classified, can the functional anomaly maps, if you sweep a threshold, classify the anatomical lesions, we find that across the group, it bends upward. Not surprisingly, based on a few slides ago, there is a correspondence between the anatomical lesion area and those hottest areas of those heat maps, and also that the lesion size correlates with the classification accuracy in each individual patient. So, okay, that's pretty simple based on what we saw. We wanted to get a little more rigorous with this question, and in this analysis, we're going to ask, can we localize in functional anomaly map data patterns of damage that we derive from the anatomical lesion data? And so we broke the brain up into a bunch of parcels. My favorite atlas, the Shen atlas, although that preference is maybe changing for no particular reason. Broke the brain up into a bunch of parcels. At each parcel, we're going to run an analysis. We're going to run the analysis by taking the percent damage to this parcel from each patient. And we're going to use that list of percent damages as a quote unquote simulated behavior. And then what we're going to do is within the left hemisphere, we're going to simulate within the left hemisphere anatomical data. And we're going to then look at just the left hemisphere of the functional anomaly mapping results data, so each patient's functional anomaly map, and we're going to try to localize in that functional anomaly map data the basically the location, the original location of the parcel that was simulated, whose behavior was simulated from the anatomical damage. So basically, in this histogram here, you can see that if we performed randomly, we would get that red distribution. On the x-axis, this is the distance between the true location of the simulated parcel and the peak discovered location of the lesion symptom mapping result using the functional anomaly mapping data. And you can see it sort of beats random chance. And we did the same thing simulating from anatomical damage and localizing within the anatomical data, which is a trivial task because it's sort of like simulating the data from your answer and then looking for the answer in your simulation, and that obviously works. But there was no difference. There was no loss in precision when we tried to go from anatomical parcel location to the functional anomaly map parcel location relative from anatomical to anatomical. Okay, fine. We're able to detect the anatomical lesion. Then comes the interesting and challenging part. What about asking the question, what is the signal outside of the lesion in the functional anomaly maps? What does it mean? Is it complete noise, which would be our null hypothesis, or is there some value and some pattern to it, like that KAP subject that we saw? And here, there's no gold standard for asking a question like that. So we thought, okay, we can assume transcallosal diaschisis is a thing. Left hemisphere damage to a certain part is going to cause dysfunction, we think, or pretty confident of, in the exact mirror right hemisphere region. And if that's the case, then what we should be able to do is use that same parcel simulation approach and simulate behaviors based on left hemisphere functional anomaly map data, and then cut off the left hemisphere functional anomaly map data and look just in the right hemisphere functional anomaly map data. And we predicted that if that was not noise in the right hemisphere, that we should be able to localize better than chance the spatial location of each of those parcels that we simulated from the left hemisphere data, which will contain the more direct consequences of the anatomical damage. And when we do that, it looks like the previous slide, lo and behold, we are able to localize those parcels with a surprising amount of precision. And of course, in this analysis, we're ignoring the x-axis, the x coordinate, because we're flipping left to right. So you can imagine taking the absolute value of that. Okay. So it looks like we're able to take left hemisphere data and find right hemisphere consequences that might match onto that potential that idea of transcallosal diaschisis. Okay, then we wanted to see, were these things reproducible? The answer here isn't so surprising on the basis of the pictures that we saw a little bit ago. When we did, we can look at the reproducibility because many patients had multiple scans. And so we did interclass correlation coefficient at the voxel-wise level for each individual patient. And across the group, if you make a box and whisker plot of that, you can see the interclass correlation coefficient is quite high. And then if we show that for each individual subject, there's no error bars when the subject only has, one pair of scans. You can see that although there's variability, it generally is pretty high. And then this pane C is just another representation of these box and whiskers, red being more reliable than the blue. There's no blue here. So these appear to look pretty reliable. Okay, so we'll wrap up with a few more slides. I've shown you how we've derived functional anomaly maps for individual patients. I tried to show that they are detecting anatomical lesions, and also that the signal outside of the lesion that we found in each of those functional anomaly maps is not simply noise, and that they seem to have very good reproducibility. So, okay, what can we do with the maps? And now functional lesion behavior mapping is going to be our example application. And here, basically all you need to know is that what we're going to do is we're going to do 4 analyses using classic behaviors, and we're going to do it with anatomical lesion symptom mapping. And then we're going to do the same analyses, but instead of the anatomical tracings, we're going to use the functional anomaly maps derived for those same patients. And so we'll predict that the functional anomaly maps should reproduce and replicate the anatomical findings, and hopefully also extend the sensitivity to lesion behavior relationships out to voxels that fall way beyond the anatomical lesion distribution, because we now have a map of not only where there's direct consequences of the lesion, but also where there's distant functional consequences. And the functional anomaly mapping results, the functional lesion symptom mapping results, will be in the hot color scale, and the traditional anatomical results in this blue-green. So we'll just look at the 4 behaviors. So on the left, you can see the functional lesion symptom mapping results. On the right, you can see the anatomical lesion symptom mapping results. And this is for auditory comprehension. We basically would expect temporal lobe like this, and that's what we see. Notably, the functional lesion symptom mapping results largely replicate what we see with the anatomical lesion symptom mapping results. So that's a good thing. That's what we were hoping to find. Quickly turning to a measure of phonological processing now, we're essentially looking at the same layout of results. And you can see in the anatomical lesion symptom mapping, we're seeing kind of, maybe call it SPT, maybe call it parietal lobe, maybe temporal lobe, but it's largely convergent with the functional lesion symptom mapping results. Turning to our speech production analysis, you're getting left frontal lobe with the anatomical lesion symptom mapping results, and that result is largely replicated in the functional lesion symptom mapping results. But you can see we also have a small cluster in the right cerebellum that is showing that the functional anomaly map signal here seems to independently relate to speech production behavior. And that's an area where in the anatomical analysis, you simply don't have any data coverage. And our last behavior here is looking at right hand pinch strength, so a motor measure. We have very strong predictions on how that should fall out. The top row here is the functional lesion symptom mapping results, and the bottom row is anatomical results. You can see in the bottom row, getting some corticospinal tract up to the hand knob here, and largely the same thing in the functional lesion symptom mapping results. It's shown translucent here because it doesn't quite reach correction for multiple comparisons, probably because there's a little break here due to the voxel resolution, but you are seeing sort of a nice hand knob here. And of course, we found it very interesting that the strongest result was right premotor cortex, contralesional to where you would expect a damage to the motor system to cause impairments in motor function. And so this, again, is suggesting that we're maybe extending the sensitivity of this type of analysis beyond the lesion boundary. And this result is consistent with what happens when, for instance, you take someone with left hemisphere damage to motor cortex and their contralesional hand recovers a bit, and then you TMS their contralesional premotor cortex, and some of that contralesional hand strength is lost, suggesting maybe that the right hemisphere premotor cortex is being relied upon to supplement that recovery. Okay, so that's our conclusion. And we can basically skip to the end here. Here we did functional lesion symptom mapping using those functional anomaly maps. We replicated classic findings, and we think that we were able to, in these analyses, extend the sensitivity beyond the regular lesion boundaries. And there may be a lot of questions about this, so we can just name the big bullet points here. So for lesion detection, we're able to have sensitivity not just to the anatomical damage, but also to functional changes outside of the tissue, outside of the lesion and spared tissue. We didn't need to segment those lesions for the functional lesion symptom mapping. It's norm referenced. It seems to be sensitive to changes in gray matter and white matter voxels. And then for the lesion behavior mapping, you can get what I'm alluding to here. That for like visual word form area, that's pretty nice. Maybe you have some more sensitivity to those regions where we don't have the best coverage usually. And then I didn't note it, but in this case, since the functional anomaly maps aren't identical, a single individual can contribute multiple times to an analysis. So some outstanding questions, right? The bold signal fluctuates. So in this analysis, we did not have a sign. So we just took the absolute value. It's unclear whether there was increases or decreases, and it's unclear what the potential mechanism is that's driving this signal. So technical improvements, scientific clinical applications, right? And this longitudinal changes, other populations. Well, this is kind of what I've been working on and what I'll continue to work on in my funded work as I start my lab. Here I'm starting the R00 phase of this grant, where we're going to be trying to deploy this type of approach to acute, subacute, and to chronic aphasia recovery to test this sort of central idea that remote dysfunction, diaschisis, which can be chronic, that resolution of that remote dysfunction or resolution of that diaschisis may relate to behavioral recovery in patients with post-stroke aphasia. And before finishing, I want to give a plug for Georgetown's T32 Neuroscience of Language training program, which is incredible. There are some slots. If you are a potential doctoral student or a potential looking for a postdoc, reach out. Sort of amazing resources. And I think with that, I'll just acknowledge my funding source, NIDCD, the Center for Aphasia Research and Rehabilitation, the Center for Brain Plasticity and Recovery here, Georgetown University, MedStar Hospital, our local CTSA. And then, of course, this is going to be like the last lecture I'm going to be able to do and show the Cognitive Recovery Lab. So this is from just a couple of weeks ago. And of course, thank you to Peter for being such an amazing mentor and like showing how to run an amazing lab and sort of inspiring us all and frankly making me believe that we can embark on this on an academic career and be successful. And hopefully the next time I talk to you all, I will have some of my own lab members to put into this box down here. And I think with that, maybe I'll stop sharing my screen and take questions.
Thank you so much, Andrew. That was great.
Thank you.
I'd like to start with questions from our room before I move to questions online. So I'm assuming you can hear us. Anything from our room? Okay. Hearing none. Okay, so then I'm going to go to the online questions. I will read those out. So Maria Ivanova says, Andrew, great research and a very nice talk. What do you think of perfusion as a measure of functional status of spare tissue? How, in your opinion, is it related to functional anomaly mapping?
Yeah, so I think this is a great question, Maria. Thank you for that great question. I think we don't know the answers to these things. We know that there's changes in the brain that are happening that allow some people to do worse, or some people to do better, or cause some people to do worse. And I think we're still learning just how to go about measuring those things. And I think perfusion is a great way to try to go about doing that. What I've talked about today, I really want to emphasize the idea that although we're using resting state fMRI, bold fMRI, I should clarify, and this support vector machine modeling approach, that I think that the specific imaging measure that we're getting, or the specific modeling approach that we're getting, isn't actually like it's profoundly important, but it's whatever works. That's the answer. And so I think we've got some perfusion data here from the past years. I think I've definitely analyzed it. It was in my K99. I saw similar patterns to what I showed today. The problem with the perfusion data that I've worked with, at least, and I guess it was sort of a P-Castle-y, the voxels were very, very large. And I think the SNR was very, very generally very, very low. And so I think what I had to work with, this is where I'm learning that you need to collect people and bring them into your lab have them carry out some of these important and interesting arms of a research program. So I think that perfusion is a great measure. The question really is, does it work? It maybe you could other people I think Dorian has done work like this, where you're basically trying to get different measures of different modalities and stack them together to try to get kind of in a machine learning sense, it might be called ensemble learning, but basically putting multiple measures of the same thing together to try to get an even better understanding of what's happening. I think the question of your question of a measure of functional status of spare tissue, I mean, I think that's an empirical biological question, but you could certainly do the exact same conceptual approach.
Great, thank you very much. And we have a question from Brenda Rapp. I'm curious to hear more about your thoughts on the basis for the transcallosal diaschisis. You probably reported, but could you remind me how or if it's related to behavior?
Yeah, so the way that we approached transcallosal diaschisis, the way we utilized it here in the study, is basically what we wanted to do is try to validate. We wanted to try to validate that the signal that we were seeing in these functional anomaly map images was in the right hemisphere, for instance, was not just random noise. And that's a tall order to ask that. And I mentioned there is really no gold standard for sort of testing that. And so we basically assumed that the idea of transcallosal diaschisis as an assumption of our analyses. So we assumed that if somebody has damage to, let's say, left BA21, that we would expect to see the exact mirror image part of the brain show some sort of functional alteration. It was kind of our way of saying, okay, we don't know what the changes in the right hemisphere might be, but the most reliable thing we could think of to do was to presume that there's such a thing as transcallosal diaschisis. I think that there's pretty strong evidence for it. Maybe that's not right, but I think that there's pretty strong evidence for it. And the way that we utilized it in our study that I described actually has nothing to do with behavior. In fact, the left hemisphere damage was the quote-unquote behavior, the simulated behavior. And we looked to localize that left hemisphere simulated behavior in the right hemisphere with the idea that we should be able to localize in the right hemisphere a location that's the mirror image of the source from which we simulated that quote-unquote behavior from the left hemisphere. But for that analysis to be valid, we had to presume that transcallosal diaschisis was a real thing.
Thank you, Andrew. I have multiple questions, but I'm going to try and limit myself. First of all, I want to know if a functional anomaly mapping is the neuroimaging equivalent to the movie Avatar. Would you agree with that, or does that fit your worldview?
I'm actually not sure. Come
Okay.
back to me. I had something for that.
I have a different one. So you started out by using basically testing the method that Julius has used before, right? Using a shell around the lesion, so perilesional, potentially active tissue, right? And you then overlaid that with what you call language potential. I forgot the term, but language viable cortex. So can you now do, or have you tried now doing the same with, instead of the lesions, using the functional anomaly maps and creating a shell around those areas, and then look to see whether, and to see if those areas overlap with language viable tissue, whether you see higher potential for activation?
You got about a half hour. I'll get back to you. That's such a fantastic, logical and focused and systematic question. I actually don't think that I have done that, but that makes perfect sense. And I think I might do it later today.
All right, we'll have you back tomorrow. That's great. Okay. I had one final, so I would really like to know if you have observed particular consistencies, so across patients, in the relation between lesions, so site or size, and particular distant functionally anomalous regions, right? So if someone has a lesion in left IFG, with the chance that a particular homologous or non-other region is also affected.
Yeah, so I don't know that I've looked systematically. Again, it makes perfect sense. I'm thinking of, Dorothy Sauer taking maybe frontal lesions and temporal lesions, splitting them out, looking for some robust, even if it's not super detailed, some general dissociation. And I'm not sure that that I've done that. I mean, again, you got about 15 minutes. One thing I can say is that there's definitely patients that show contralesional, like completely wild signal. And if we look at our data, I mean, I think, forget functional anomaly mapping or whatever for a second, but like, I think if we look at our data carefully, there's stuff in there, both around the lesion and far away from the lesion, in particular, contralesional. And what those things mean, I don't know. I haven't. It's inconsistent, right? That's one of the challenges. There's the heterogeneity.
Right. Okay. Thanks a lot. Those are the questions that we had online. We had no questions, further questions here. All right. Thank you so much, Andrew. That was great. Good to see you. And we hope to see our audience in 2 weeks' time. Thank you.
Thanks so much for listening. I appreciate it.